So thank you everyone for joining. It's great pleasure to introduce our speaker today, Dr. Xin He. Xin is currently an assistant professor at Department of Genetics at University of Chicago, although he has been approved to be an associate professor, and so he's tenured already. So congratulations, Xin. Shane got his PhD at the University of Illinois Urbana-Champaign under supervision of Saurabh Sinha, my previous advisor. So actually Shane and I, we overlapped for a year and a half or 2 years. I remember that I was looking up always to Shane. He always like, come to all of these seminars with 3 papers in his hand, looking down, and then he was never looked up in any of these seminars. And then he was asking the most profound questions and then keeping the people on their toes. So I definitely learned quite a bit. Then he went on, did 2 postdocs, one in UCSF and one in CMU. And then of course, all of them were extremely productive. Also, his lab has been extremely productive. His publication have been in prestigious journals, Nature Genetics, Nature Science, and so on. I don't want to take more time. Shane, please go on, share your slides, and welcome.
Okay, thanks, Majid, for the really generous introduction. Let me share my screen. Okay, let me put in the slideshow mode. Can everyone see my screen?
Yes.
Did it move?
It does move as well.
Okay, great.
And then if you have your point, if you have a laser pointer on that, that would be perfect.
Do you see my drawing?
Yes, we can see that you're writing on it.
Okay, great. All right, thank everyone for coming. So I guess the topic I'm going to cover today is a little different. So I'll give some background. Hopefully you'll learn some human genetics from my talk, and feel free to ask questions during my talk. So the title of my talk is using Mendelian randomization to infer causal relationship of complex traits. Yeah, so my lab focus on human genetic studies. So I'll give a little bit of background about this about human genetics. So we know that everybody is different. Everyone is different in their appearance, in their physiological trait, their height, body weight, and so on. And people have different risk of disease. So human genetics is basically about finding DNA variations that explain these phenotypic variations. So why do we study human genetics? Because essentially, human genetics can provide insights on diseases and can inform drug discoveries. So the figure shows a possible basically path from genetic studies to possible therapeutic advance. So from the left, you can see this. This shows the result from a typical association studies. So I will talk about it in a minute. So those basically see which variants, DNA variant, the genome associated with the risk of disease. So from these association studies, you can identify some genetic or DNA variants, and then you can study their. This gives you a clue about what are the important mechanisms that may lead to disease. So you can then you can then study the possible mechanism. You can say, what are the downstream effects of the DNA variations? For example, you can study their how gene expression associated with the genotype changes, right? So as you see in the middle. And then you can study the physiological consequence of genetic variations. For example, the variant might increase your neurodegenerations, right? And these studies will basically help you to come up with a list of drug targets. And basically the idea is that if you can introduce a drug to mimic the effect of genetic change, you may be able to reverse its phenotypes, right? So, so there's many examples of drug targets that are informed by human genetic study, by human mutations. For example, cystic fibrosis is a pretty severe, we call Mendelian disorders, is often run in families. So genetic study implicates gene CFTR, and drugs have been developed to target the CFTR gene and shown to have good effects on cystic fibrosis patients. So another example is heart disease. So heart disease is really complex disorders, as it doesn't involve a single gene, involve hundreds of genes. But still, people find that a gene called PCSK9, this gene can significantly reduce the lipid level, the LDL level, which is a major risk factor of heart disease. And there are many drugs being developed to target this PCSK9 gene. So hopefully that's kind of motivate, give you some idea why we do these studies. So the main approach to map this genetic variants of disease is called genome-wide association studies. So so in GWAS, so this slide gives you some ideas of how GWAS is conducted. So in GWAS, you typically start with collecting DNA samples from cases and controls. So cases are individuals that are affected by the disease, and controls would be healthy individuals, but they match cases in their gender, their ethnicity, and other aspects. So, and then you can sequence or genotype your DNA variants. So this one shows a microarray based genotyping. So you can so each spot in the array basically shows the genotype. So you have basically different colors, means that the case control have different genotypes. So you can then compare the frequency of variants, so frequency mutations in cases and controls. For example, if variant, say if A2T mutation is say 30% in cases, but only like 10% in controls, then you know that the variant is associated with the risk of disorder. It's overrepresented in cases, right? So that's give you what we call associative variants or genetic associations. So the results often are visualized by this Manhattan plot. So it shows the statistics of association of all these variants in the genome. So y-axis shows their minus log10 p-values. So if the number is usually, if it's less than something like around 10 to the minus 8, with really small p-values, then those are significant associations. Right, so that's what is typical GWAS study. And one thing you can notice that in the GWAS, often you got the peaks, but the peak contain not a single SNP, but it contain a bunch of these single nucleotide variations, single nucleotide polymorphism, the SNPs, right? So as you can see in the red circle here. So those are variants that are close to each other. So in human genome, basically the variants, if they're close to each other, they are often in what is so-called linkage disequilibrium. Basically means that those variants are highly correlated. And those variants, basically the variant come from a common ancestor. And this variant generally would have similar associations with the same phenotype because they are highly correlated with each other. So that's why you see this kind of peaks, which often has not one variant, but multiple variants, could be tens or hundreds of variants with similar association statistics. Okay, so that's like 1 minute introduction of GWAS. Now, GWAS have been highly successful in mapping genetic basis of complex traits. So, so this slide shows the results of schizophrenia. So schizophrenia is a complex mental disorder. The figure shows the progress over the years. So the red bar shows the number of loci that are implicated. So and so that's red bar. The blue bar shows basically sample size, measured in terms of thousands of samples. So as you can see here, in like 10 years ago, it's 2009, we only know like a couple of loci associated with schizophrenia. So we don't really know why some people have schizophrenia. We know very little about genetic basis, even though schizophrenia is highly heritable. It's often run in families. But in 2014, we have more than 100 loci implicated. And in the upcoming study, the PGC3 study, more than it's close to 300 loci have been implicated in schizophrenia. Right, so this has been highly successful. However, it's difficult to translate these GWAS findings, GWAS associations into like mechanistic insights or therapeutic advance. And the reason is that most GWAS variants have small effect size. So they have pretty small impact on the phenotype of the patients. So for example, this slide shows the result of GWAS of height. So each dot represents a SNP, and x-axis shows their minor allele frequency of the SNPs, the single nucleotide polymorphism, and y-axis shows their effect size on height. So measured in terms of minor allele effect size. In other words, if you have this variant, how much height you wear, like what is the effect on the height of the person carrying that allele? So it's measured by centimeter. So as you can see, only in rare cases, a single mutation or single variant increase or decrease height by more than 1 centimeter. In the vast majority of cases, effect size is far less than 1. It's just something around like 0.2, 0.1, right? So the effect size is really, really small. So having this mutation, like on average, increase your height or decrease your height by like 0.2 centimeter, right? So it's really, really small effect size. So that makes it harder to kind of know biology. And another challenge of GWAS is that in most loci, the causal variant are unknown because of linkage disagreement. So that's the point I made in the previous slide. And because you don't know the causal variant, often you don't know what are the genes, right, that are disrupted by the variants. So that makes it difficult to identify, like, say, the drug targets. So one idea to address is to challenge the small effect size and the lack of knowledge about the causal genes. So one strategy to address that is to focus on what we call the intermediate risk factors. So the kind of conceptual model is that there's many genetic variants can increase or decrease the risk. So it's showing the top here. So it could be hundreds of, in some traits could be thousands of them. But in order for this variant to affect the disease, they have to affect something in the middle, right? And the things in the middle could be genes or the metabolites, or they could affect the number of cells, right? The cellular compositions or the kind of morphology of the cells. And they could also affect physiological traits, for example, your blood pressure, right? So it's this intermediate risk factor that eventually act on disease, right? And those risk factors essentially immediate effects on many variants. So if you can identify this intermediate risk factor, that can really help us. It can help us explain the effects not of single variant, but multiple variants, right? Because the multiple variant might affect on single risk factors. And also these risk factors, once we identify them, can become a drug, like a treatment target. So one well-known example is low density lipoproteins, LDL. So it's a major risk factor of heart disease. And there are many drugs developed for targeting or reducing level LDL, right? So if you can identify the risk factors, can really help us to reduce the risk of disease. And we are basically use human genetic studies using the genetic variants to identify the risk factors. So that's a topic of my talk. Okay, so let's say we have a potential risk factor, let's say lipid level, LDL or HDL, and we want to evaluate whether this risk factor have effect on a disorder, say type 2 diabetes. Okay, so lipid is a risk factor for heart disease, cardiovascular disorder. So it might be a reasonable hypothesis to say that lipid might affect risk of T2D, right, which is another major common disorder. So you might want to say, can we just say measure the lipid level of many patients and say whether the lipid level associated with the risk of type 2 diabetes, right? So, but as any scientist, we know that the correlation doesn't mean causations. And the reason is that there are a lot of potential confounders. So for example, diet. So if you eat heavy kind of high fat diet, you might imagine that could increase your lipid level, and it might also increase your type 2 diabetes risk. So this would be one possible confounder of the diet, and other things would be exercise, like how much you exercise, or smoking. Those things can affect both traits, right? So, so in statistics, when you have confounders, often you can do regression analysis and adjusting for confounders. Basically, you put confounder as a covariant in the regression model. So the problem is that there are many potential confounders. Even if you may not be able to measure all of them, even if you measure all the kind of conceivable confounders, there may be more that you haven't measured, right? And that makes it really harder to translate the correlation into the causal effect, right? Because there's always the risk that some confounders are not measured, that are creating correlation between your risk factor and your disease. So how do we deal with that challenge? So and the gold standard of the field is using randomized clinical trial. So this slide explains the idea. So suppose we have a drug, okay, and we have treatment. We want to evaluate the effect of the drug, whether the treatment can have any kind of effect, causal effect on the outcome. So the way you solve this problem is that you basically have sample population. You basically randomly partition your subjects into two groups, right? So group 1, group 2, group 2, and then for one group you have the treatment, and for the other group you put a control in there, sometimes called placebo. You basically put a control. You don't really treat them. You just put some kind of controls, and then you compare the outcome. So basically, if you have, say, lower disease risk in the first group, then you know that that difference must be due to treatment, because the two groups are identical in every aspect. The only difference is that the first group received treatment, right? Because patients are randomized and randomly partitioned or assigned into two groups, right? So the distribution of the two groups of the patient would be identical. They'll be same in age, in gender, in everything you can think of, because it's totally random assignment, and the only difference treatment, right? So that's a kind of gold standard approach to establish some treatment has some effects. So this randomized clinical trial or randomized treatment can be used to study not only drugs, but essentially any potential factor of interest. Okay, so imagine you can do that for our problem. So you can imagine, like, instead of doing human study, you can do mouse study, and then you can imagine you do randomized treatment on lipids. For example, feed them diet. Certain diets increase their lipid level, right? So so in that case, you create a randomized clinical trial. So the important idea is that this randomized treatment would be totally independent of environmental exposures. It's confounders, right? Because it's totally random. And it would be totally independent of the outcome, right? Because you treat them essentially before you observe outcome. So this kind of diagram, this kind of causal diagram, so each edge represents the potential causal effect of one variable on the other. So you basically don't have the link from treatment to type 2 diabetes directly, or randomized treatment to the confounders, right? So the fact you don't have those links basically helps to establish causality. So that's basically randomized clinical trial. So the idea of Mendelian randomization is to use genetic variants to mimic randomized treatment. So that you can see in this figure. So the idea is that we might have some genetic variant that perturb lipid level. Okay, so as you can imagine, lipid level in the body is controlled by many enzymes, by many biological pathways, right? So if you have a variant that, say, increase or decrease the level of enzyme, that might increase or decrease the level of lipid, right? So let's look at one variant at a time. So that variant has two alleles. One allele leads to high lipid, one allele leads to low lipid, right? So then you can imagine that you can partition all the individuals into those carrying the high lipid allele and those carrying the low lipid allele, right? So now you have two groups of individuals, just like randomized clinical trials, right? So each group carrying a distinct genotype. One leads to high lipid, one is low lipid. Okay, so it's like a treatment. And we can argue that treatment is randomized. Basically, which allele a person inherits is determined during conception, right? It's determined during the reproductive process. And of course, that is independent of your later environmental exposure, right? Diet, exercise, and so on. And that of course it's independent of your risk of type, whether you develop type of diabetes later in your life, right? Because so that's giving you the name of the Mendelian randomization. So Mendelian randomization basically means the segregation. So in the mother or father, we have the mother carries two alleles, say high lipid allele and low lipid allele. Which allele enters the child will be totally random, right? It's Mendelian segregation. So that's give you the name of Mendelian randomization. Okay. All right. So that's basically the idea. So I'll give a little math to show how this idea can be used in practice. So basically, we have a trait, mediator trait. So that would be kind of lipid level in our case, and the outcome. So this would be risk of disease, is whether you have say diabetes or not. And we have this causal effect gamma, which is unknown. So there are potential confounders, diet, smoking, and so on, acting on the both mediator outcome. And we have variant G. So in this case, we choose variants that have effect on mediator, right? You can think about this variant that increase lipid level, for example. And our goal is to estimate gamma. But we have some data. We have these statistical associations of variant G with mediator M and variant G with respect to the outcome Y. Those are the GWAS studies, the association studies I introduced earlier, right? So I have this beta M and beta Y. So we use beta M to denote the effects of variant G on the mediator M, and the beta Y is effect of or association of G with outcome, right? So those things are obtained from the association studies. Now we can write this equation. So the simple equation. So we can relate this gamma with the mirrored associations. So basically we can use delta G to represent the difference of genotype between between any two groups, right? So usually we encode genotype as 0, 1, or 2. So if someone is basically reference genotypes, carry 2 major allele will be 0. If carry 2 minor allele is it's 2. If you carry 1 minor allele is 1, right? So you can encode the genotype numerically, and the difference of genotype between two groups we call delta G, and difference of phenotype we call delta Y. Okay, so the ratio of delta by delta G is basically the effect of variant G on the outcome, and that's just beta Y. And you can rewrite this. You can rewrite this as delta Y divided by delta M. So that's like two groups of individuals that differ by genotype. You can measure the difference of M, the mediator, equal delta M. And the delta Y divided by delta M, that's basically effects, the causal effects, right? That's basically you say you will change mediator, you change your risk factor, how much change you have in the outcome. So that's gamma. And delta M divided by delta G is just association or the effect of variant on the mediator to beta M. So you basically have this very simple linear, this very simple regression, very simple model. So the effect of variant on the outcome is effect of variant on the mediator times the causal effect of mediator on the outcome. Okay. Now you can see that in genetic data, you measure beta Y and you measure beta M, you just take the ratio and you can estimate the causal effect, right? So it's very, very easy, right? The whole point is if you have variant that perturb M, and then you can measure association of G to M and G to Y, and you take a ratio, you get a causal effect, right? So it's extremely simple. So so this regime can be generalized. So sometimes it's easy to use multiple in variants. So this variant called instruments. So you can use multiple, assume that this assumption holds, the assumption that, so the key assumption is that G, this variant, doesn't act on confounders, it doesn't affect on outcome. If this assumption holds, then you can basically write down this equation, right? Because this equation holds for every variant, right? Then you can basically do a regression, right? So, so your response variable is beta yi, and your exponential variable is beta mi, right? So the I is the index of variant, and do regression. You fit a linear line, you get estimated causal effects. So it's really easy. And often people use strong instrument. In other words, they use variant have really big effects on M to do this inference. And sometimes you have a countable estimation error of effects. What that means is that the beta M and beta Y, those come from association studies. Those have some measurement errors because yeah, it comes from a statistical test. So there's some uncertainty of the true effect size. So the standard approach of fitting this multiple essentially this regression model is called inverse variance weighting. It's called IVW. So I'm not going to talk about details. So this is one example of how MR can be used in practice. So this shows that MR supports a causal role of LDL cholesterol in coronary artery disease. So x-axis show, so each spot dot here show the a SNP of genetic variants that's associated with LDL cholesterol. So people have done GWAS of LDL and get the variants that are associated with LDL level. And similarly, you can do GWAS on CAD, and you can get an effect size also of variants on CAD risk, right? So then you can plot the effects on LDL versus their effects on CAD. And the size of the circle represents the strength of association with LDL. So large ones really means really strong associations. Now you can see there's very nice linear lines, right, of these two effect size. Right, so x-axis is the beta M term and y-axis is the beta Y term, right? And the slope would then basically be gamma as we talked about earlier, right? So as many of you probably know, the LDL cholesterol is known through many studies that is actually a risk factor for CAD. All right, so that's kind of the background of MR. But the challenge of MR to use MR in kind of in more general settings that these assumptions are often violated. And these violated variants have what we call pleiotropic effects. So what it means that our key assumption is variant cannot affect its confounders directly, and the variant cannot affect Y directly without going through the mediator. So if this happens, let's imagine like the variant acts on something else which affects, say, type 2 diabetes risk. If this pleiotropic effects happens, that violates MR assumption. And it is created this non-causal paths. So in other words, G can affect the U, which affect Y, or G affect Y directly, okay? So when this happens, this will be problematic. So just imagine the variant, let's say, variant affects Y directly. In that case, your association of G with Y doesn't reflect anything about gamma, right? Because it's coming from different paths. But you're still fitting the rational. You're assuming that beta Y term is proportional to gamma, right? You're making that assumption in your model. But that assumption is wrong because G can affect Y directly, right? So that association doesn't tell you anything about gamma. Then your estimate of gamma would be wrong. So so that's kind of the major challenge of the field. And the problem is that, often these genetic variants often affect a lot of things. So this pleiotropic effect is actually quite common in human genetics, right? So for any particular trait mediator M, you don't know whether the variant of M affects something else or not, right? So in real case, extremely hard to verify the assumption of MR, basically. So there's some method we developed. I'm not going to talk about the details, but basically a lot of times this method is trying to remove these outliers. So outlier basically means a variant that acts on Y directly at a pleiotropic effect. So the method developed to remove this kind of variant valid assumptions. But however, so all the current methods do not explicitly address this problem of hierarchical confounders. So I'll talk about what that means. So hierarchical confounder basically, so we are thinking about a different scenario. So in the previously, our confounders are like smoking, exercise, and diet, right? So it represents like your kind of environmental kind of variables. But imagine your confounder would be something like this: BMI, the body mass index. So BMI is a known risk factor of T2D, and BMI has probably broad effect on your body, and you can imagine that could affect lipid level. And another thing is like smoking. So smoking, even though smoking is a kind of non-genetic trait, but smoking depends on addictive behavior, right? Whether people it's kind of prone to addiction, right? And that behavior trait is often highly genetic. So that's well known, right? So in this case, these co-founders, they're more complex. They're not just environmental exposures like smoking. They're actually genetic, like they actually can be affected genetically. So that's what we call heritable co-founders, right? So in that case, you could imagine there's a lot of genetic variants would act on this BMI or this additive behavior. It's genetically determined the co-founders. Then what happens is variants acting on the co-founders would have correlated effects on both the mediator, the lipid in our case, and the outcome, right? So that could create a problem because that creates essentially correlation of genetic effects on lipid and genetic effects on diabetes that would create correlation across many variants because all these co-founders I described here are what we call polygenic. So in other words, there's many variants that can potentially affect BMI or additive behavior, right? So to give you some sense, so let's look at, we have done some simulations. So in this case, we have a mediator, a potential risk factor M, and its outcome Y. So there's no causal effect, but there is a heritable cofounder U acting on those M and Y. So we have a lot of variants acting on M, but most of them, they don't affect on U, 85% of them. But 15% of variants act on M, they actually act on M through this unobserved cofounder U, okay? So you can think about U as a BMI. So in other words, let's say, imagine BMI acts on lipid, some variants basically act on BMI, which in turn act on lipid, right? Those variants will be associated with lipid, right? But there are other mechanisms that genetically affect lipid level. So you got 85% of variants don't affect on the lipid level, right? So that's the situation. Then you can basically, we can do some simulation to simulate, under this model, you can simulate the effects of variant on M and Y. So for black ones, so the variant coming from the first model, you see there's no correlation as you expect, right? So, but for the blue ones, because the variant acts on the U and U affect on both M and Y, so you see a correlation, the blue ones. The challenge is you don't know which variants can affect on U or not because U is unobserved. So if you fit a standard MR model, you will see a good correlation. You got p-value 0.01, right? So that leads to false positive by MR. So that's the problem that we are trying to address in our work. So our idea is basically we are relaxed assumptions of MR. We allow variant to have pleiotropic effects. So, so we basically allow two kinds of pleiotropic effects. First, in most of cases, the variants kind of valid instruments, but not valid instruments. Most variants they don't affect on the unobserved confounder. They only affect Y directly. Okay, some of them may or may not affect Y, but the point is these pleiotropic effects would be uncorrelated across different variants. Okay, so this is the most variants. We call this uncorrelated pleiotropic. Second scenario is more complex. This is what we call correlated pleiotropic. It's a variant effect on U. And because it's variant effect on U, their effects have to be correlated across different individuals. So if your variant has a big effect on U, then it will have a big effect on M and Y, right? If it has a small effect on U, it has a small effect on M and Y, right? So those pleiotropic effects will be correlated across individuals. So, and for our purpose, we call this U as a shared factor. This shared factor U captures essentially how genetics of M and genetics of Y are kind of correlated across variants. So we have these two kinds of variants, just like what I model. Yes, so that's basically what I talked about earlier in the simulation setting. So we have these two scenarios, and so in practice, because we don't observe U, so we don't know which variants belong to which scenario. So we are introduced indicator variable to model these two kinds of effects. So these two kinds of pleiotropic effects. So Z equals 0, basically it means uncorrelated pleiotropy. So the variant might affect Y, but doesn't affect on cofounder or the shared factor. The second case, Z equal to 1, the variant acts on the shared factor. So Zi follows the Bernoulli distribution with mean on Q. That's our assumption. So the Q kind of mirrors how often, essentially, it mirrors how often their effect size of M and Y are correlated. So for the variant not acting through the shared factor, the model is pretty simple. So in this case, we have GI effect on M. So the beta MI is the effect size of GI on M, and then causal effect is gamma. And we potentially have a pleiotropic effect of GI to Y directly, we call this theta I. And then we have this kind of regression model. So, so the effect of variant on phenotype beta YI now has two parts. The first part comes from causal effect, right? So that's we talked about earlier. It's basically GI effect on M and effect on gamma, and M effect Y. So it would be causal effect gamma times beta MI. So this is causal effect we talked about earlier. And then we have to add this additional term, is theta I. Theta is a direct effect. So theta is unknown. So we use a we basically assume theta is sparse. So biologically means that for most variants, this effect is probably zero, but occasionally this effect is non-zero. So next, we are model variant X through the shared factor. So this one is a little more complex. So basically, so we assume that now there's a shared factor U. So U could affect M and affect Y. So we assume the effect of U to Y is eta, is parameter that we don't know. And we assume U effect of U to M is 1. So the reason we set this 1 is basically U is unobserved. So you can arbitrarily scale U, right? So you basically divide you can arbitrarily scale U so that the effect is always 1. So that makes math somewhat simpler. It's just because it doesn't introduce additional parameters. It eliminates one parameter. And then we call GI effect of GI to U as beta MI, right? Because the reason that we can, the GI to U effect is just the effect of GI to M is the effect of GI on M is just the effect of GI on U times effect on U to M, right? Because we set this as U, set this U to M effect as 1. The effect of GI to U is just beta MI. Okay. And we still have this additional pleiotropic effects. So the basic idea now is that this beta YI term has 3 parts: the causal effect, right? So the GI can affect Y through this causal parts to go through U and M and Y. So this is the same as before. Now we have the additional term. Now we have this possibility that GI affect on U and the U acts on Y, right? So GI to U and U to Y. That's another possibility. And that effect is GI to U effect, which is beta MI, and the U to Y effect, that's eta, right? So this additional pleiotropic effect, GI affects on U, U affects Y. So the second possibility, second parse, the GI can affect Y. And finally, we have this correlated pleiotropic effect, theta I. So now we have 3 parts, and we have kind of extended regression model. Now we can put these models together. So we call this model cause for causal analysis in summary effect size. So we can write a single model. The single model is basically the same as before, except that we have this additional indicator variable there, right? When ZI equals 0, the second term disappears, right? That's the first case. When ZI equals 1, it reduces to the second case we talk about, right? So it's basically a regression model of YI effect of variant on phenotype and effect variant on the exposure on the mediator M. It's basically a regression model, right? It's somewhat different from the standard regression model in that it's a kind of error term first, and secondly, it has this kind of unobserved hidden variable ZI. Okay. So, but basically, it's kind of regression model. So before I talk about kind of the inference, statistical inference, I give some intuition how this cause model works. So basically, we have this causal model where the variant acts on phenotype. Sorry, the mediator or the exposure acts on phenotype as a causal effect. And second model is the sharing model. So sharing model basically says that the variant has no causal effect. The mediator has no causal effect on the trait. but the variance can act on the unobserved confounder U. Okay, so the sharing model basically gamma equal to 0. So it corresponds to the case where gamma equal to 0. So that's sharing model. So our primary goal is to test whether gamma equals 0 or not. Because gamma equals 0 is causal model, right? There's a causal effect. Gamma equals 0. If gamma is not equal to 0, that's causal model. If gamma equals 0, that's sharing model. So in that case, there's no causal effect, but the two traits M and Y may share some genetic basis. So if some confounder can affect them both. So the basic idea that we can distinguish two models is that under causal model, whenever the effect size of variance on M and effect size on Y would be correlated. So if variant perturb M, then the variant would have corresponding effects on the phenotype. So you'll see many variants fall into the green lines here, right? That's what we expect from linear regression. And there could have variant act on Y directly without going through M. So that's possible to the orange line, because the phenotype can be affected by many other things, right? So you basically your data would follow these two lines, two kind of ellipse. So if on the sharing model, what happens is that you could have variants acting only on the trait M, but not on phenotype. So there's the red lines here, right? So the variants can change your M, but because M doesn't have a causal effect on Y, right? They may not have only effect on Y, so they will basically be close to the x-axis. So the red ones here. You could still have green ones because those are variants acting on the shared factor, right? So the variants acting on shared factor would have correlated effect on M and Y, right? So on the sharing model, you basically have the green one, red one, and orange one. And the intuition is the ones in the red parts, those red variants, those are variants that we that helps us distinguish these two models. Okay. All right, so and I'll talk a little bit about the inference challenge, and then I'll talk about the results. So this is a model, and the model has 3 parts, as I talked about earlier. So the problem is beta mi and beta yi term, they have observation errors. In other words, they come from measurement, so they have measurement errors. So usually to model this measurement error, use a normal model. You basically assume that the measured effect of SNP I on trait M on the mediator comes from normal distribution with mean beta mi as a true effect and standard error. So your data is actually beta hat and S hat, but you don't know the true effects. Right? And similarly, you have this measurement error for the association of SNP I with phenotype Y, and it also has measurement error in there. So basically, yeah, this term is unknown. Beta Y is easier because beta Y depends on beta mi. If we know the parameter, we know beta yi. So that's one thing I want to point out. And secondly, I want to say that many variant is small beta mi informative. So as I mentioned earlier, in GWAS, often the trace, people find that many variants have small effect size. So many variants have small effect size on mediator M. But they could be informative for our purpose. They're often ignored in the standard MR setting. So, and the next variable parameter is theta I. So we don't know the theta I term, and Zi is unknown, as I said. So so there's a number of unknowns, right? So beta mi, theta and Zi, right? So these are a lot of unknowns, and if you use many variants, then there are many parameters, right? So the general idea when you do a statistical model, have many unknown parameters, but you're not interested in that. So you're interested in gamma in our case, the causal effect. So in this case, use a Bayesian inference. Okay. Now, one parameter you really want to know about, but there's a lot of other unknown parameters you want to get rid of. The way to do that, you marginalize. You trace the parameters as random variables, and you take integration. You integrate out this thing. So this is the idea of Bayesian inference. So the data beta hat is all the data we have. Gamma is the main parameter of interest. My model is defined in terms of true effect beta and theta, these unknown parameters. So basically, I write this probability in terms of this probability, but you integrate out this unknown parameter beta and theta. Okay. So this is the idea of Bayesian inference. And then for this unknown parameter beta and theta, we assume is from a mixed the prior distribution. So we use a mixture of bivariant and normal distribution. So which is pretty commonly used when you have two parameters are somewhat dependent. They use normal distribution, right? Use bivariant normal. But in our case, we use a mixture of normal distribution. So the main reason is that we want to model some sparsity. So which is common, I think for people who have used learned machine learning, you know that like lasso kind of model, right? So you basically want your parameter to be zero most of the time. So in Bayesian statistics, you can do something similar. You basically assume the prior distribution of the parameter as zero most of the time, right? So we use a sparse distribution. So basically use pi now is probability of the parameter zero times this delta zero. Now delta is basically a function that is a distribution that is always zero. And the rest is a mixture of bivariant normal distribution. So I'm going to skip the details. But basically, we have this four model. We have likelihood function of our data, and we have prior distribution. So the prior of zi, this is indicator variables for the Bernoulli distribution. So as I talked about earlier, and the parameter for the mixture of normal distribution. And then you can, once you define this distribution, you can statistically integrate out those parameters and to infer gamma, right? So, and primarily we are testing whether gamma equals zero or not. So we testing this, we compare the causal model where gamma is free parameter versus a non-causal model where gamma equals zero. We just compare this model. So that's kind of like hypothesis testing problem. We use something called ERPD to compare the two models. So I'll skip that. Okay, so that's our model. So we first assess the model using simulations. So we found that this cause reduced false positive rate in simulations. So so basically in the simulations we have no causal effect. So gamma equals zero, but we have this Q. Q is the proportion of variant acting with the shared factors, right? So we vary this this Q parameter from 0 to 0.5, and y-axis shows how often the method detects significant effects. So those are basically false positive rates. So we compare Radovan cause, that's our model, and some other some other MR methods. So IVW is standard MR. So these two methods is basically trying to control for these pleiotropic effects, but they don't explicitly model the hierarchical confounder. So we can see in most cases, the cause, the Radovan have very low false positive rate, where other methods have high false positive rate. So in the next, we are looking at the, we introduce causal effect. So in this case, we have both non-causal effect and causal effect. And we ask the method to kind of classify, it's a kind of classification problem. And then we show the result using this ROC curve. So you can see that cause is much better at, the red one is much better at discriminating causal and non-causal effects than other MR methods. And then we apply this causal model, to study to the real data. So we assess possible causal effect of common risk factors on the phenotypes. So for common risk factors, we look at the body mass index, smoking, blood pressure, lipid level, fasting glucose. And for the phenotype, we look at a few common disorders: asthma, CAD, type 2 diabetes, and stroke. So because the challenge for the real data is we don't know the truth. We don't know whether a pair of risk factor phenotype has causal effect or not. So we did a very comprehensive literature search, and we classified these pairs as a known causal effect. Some literature support, but unclear or unknown, implausible. So in general, we find this method agree for the known case, and the literature support case also somewhat similar. But in the category unknown implausible case, cause find only 4, where other methods find a lot more. Okay, so we believe that most of the last category probably false positives. So so as one example is the ARDR and T2D risk case. So in this case, All the other methods, IVW, MR Presso, and weighted mode show pretty strong p-values. So we plot this effect size of variant on LDR and the effect size of variant on T2D. So as you can see here, there's kind of overall an active association. So that's why this method find all these other MR methods report strong p-values. But of course, doesn't report p-value. There is a p-value of 0.3. It's very insignificant. And the reason is that to understand why, so we plot the SNPs by their contribution to the test statistic of a cause, right? So each variant basically makes some contribution to the cause model. So the purple ones are the ones that support the causal model. So as we see, the ones that are close to the lines would have purple one. So it's basically support the causal model. But of course, find a lot of variants with green ones. So green variants, variants colored in green are variants that reject causal model. So you can see some of them to understand, give some intuition. So let's say, look this one, the ones with really large green one. These are variants basically have a large effect on LDR that has no effect on T2D, right? So this one heavily contribute to the statistics of cause. That's why our model reject the causal model, right? And there's a bunch of other green ones, and there's probably a lot of green ones here that you really cannot see here, right? So the intuition here is that you kind of have a lot of variants that essentially mimic preservation of LDR, right? But you don't see any risk of T2D, right? So that's really the evidence against causality. That's why cause comes with this conclusion there's no causal effect, okay? So that's going to hopefully give you some idea. And in this case, the truth is unknown. We don't really know, but at least I think the result of the disagreement of cause with other methods essentially kind of motivates the kind of the idea that the result from earlier MR result may not be valid. They may be susceptible to the false positives. All right, so we assess the use other ways to assess the result, which is kind of in those cases we kind of know better what are the know the truth. So we first evaluate the reverse causality. So we treat disease as risk factor and the risk factor as phenotype. We kind of switch the order of testing. In this case, we don't expect disease to affect risk factor, right? We expect the other direction. So in this case, if you find a significant result, it will be false positive, right? Because we know biology goes in other way. So we plot the result of many all these four methods. So the size of a triangle shows the strength of statistics. So if it's solid, it means its result is statistically significant by a certain method. So as you can see here, cause in general avoids the reverse causality. There's a few cases, and essentially there's only one case where cause reports strong association, CAD to SBP. But in most cases, we avoid this reverse causality, where other methods report more. So in particular, as you can see for CAD case, the IVW and this method report a lot of essentially false associations. So another way to assess result is we use a collection of blood cell trait as a risk factor. So those are numbers or properties of blood cells. So for example, the morphology, the size of your red blood cells, right? Or white blood cells or platelet cells, right? There's a bunch of immune blood cells. We treat them as risk factor, and we study some immune disorders, autoimmune disorders, and SLE, IBD, and RA, and allergy and asthma, right? Because these are immune disorders, we expect some of the blood cells would be may have some causal influence on this disease. But the important thing to know is that even though these three types of blood cells, they come out from same ancestors. The same progenitor cells generate all these three types of cells. So their genetics actually are known to be correlated. So you know that often one variant acts on red blood cell count would also influence white blood cell counts. So that's known. So the nice thing about this system is that even though the genetics is shared, we know only white blood cells should have causal effect on these disorders, because what red blood cells, they don't have immune function. And similarly, platelet cells, they involved in blood clotting, they also have no immune functions, right? So it's a good system where we kind of know what are the false positives, what are the potential true positives. So we evaluate this system and it displays results kind of as in the same way as before. So each row shows one mediator, one exposure, and each column shows one immune disorders. And we group this trait by platelet, red blood cell, or white blood cells. As you can see here, we don't really expect, we shouldn't see causal effect on the top two, because those are irrelevant to immune disorders. And cause basically avoid almost all the false positives there, except in one, right? Where other methods report a lot more false positives. So in the bottom, in the red blood cell case, cause does find 3 potential causal effects involving this cell type called the eosinophil. And eosinophil basically is known to play important role for allergic disorders, for allergy and asthma. And it's people actually explore eosinophil as a therapeutic targets. So those are true positives. So the role of EORA is unknown. Right, so this result hopefully gives you the idea that we kind of really avoid, most of the time avoid false positives, where or what would be shown as causal effect are likely to be biologically true. So last one, the summary. So we show that current methods are prone to false discoveries because of the hierarchical confounders, and causal model counts for potential confounders. And essentially, I didn't speculate, different emphasize too much, but basically, cause can use many instruments to make inference. And we show in both simulation and real data, cause is better at reducing false positive findings. And this paper is published in Nature Genetics last year. So the work is done mostly by my former postdoc, Jim Morrison, and my student, Nicholas, and it's project co-supervised with my collaborator, Matthew Stevens. Okay, so with that, I'll stop. Take any questions?
Thank you so much, Shin. That was a great talk. So I know that we have run a little bit out of time, but we can take a few questions. Is there any questions from audience?
Actually, I wanted to ask a couple of questions. This was very clear. It was an explanation was very clear to me. Thank you very much. Actually, sometimes when I attend the statistics talks, I'm lost in the middle of the talk, but this was very
thank you.
You explained really nicely. So in page 22, I have a couple of questions. I just write down the questions I had. In page 22, you had the eta, which is how do you call it? This U, yeah, which yeah. So here in this diagram, there's only one U, but I think in reality there will be lots of those how do you call it, this shared factors. Maybe G may be affecting to lots of U's. So in that case, you will have lots of etas.
Yeah,
can you consider that, or is it like you summarize everything and then?
Well, that's extremely good comment. So we are thinking about that. We're thinking to a lot more shared factors in a more general model. So that's something we have thought about. And we didn't use that in this model, and that may not be a big, huge problem, because U cause a problem only when it causes correlation of effect size, because that's be similar to a causal effect. What do you expect under a causal effect? If you have many factors that kind of cancel out each other, you don't generally expect the correlation. So even though you kind of miss something, it doesn't cause false positives. So that's basic intuition. So capturing the correlation due to non-causal factors would be main issue. So that can be done by using single factor.
I see.
That's why, yeah. But that's very, very good point.
Okay. And I had another question at page 31. I don't remember what it was.
Actually, Daisuke, can I ask one thing?
Yeah. Oh, sure.
Because it was in the same slide. And then we'll go that, because this was very good point you brought up. So Shin, if we were, let's say, having multiple confounding factors, and then the model could, and then those are your hidden variables to some extent, right?
Yes.
And then if you are learning those hidden variables, do you think that we could potentially identify a confounding that we have not considered, or associate those confounding factors with known factors? So let's say, can we go back and say, oh, maybe this confounding factor is smoking, this confounding factor is X and Y and Z?
Yeah, that's another very good point. Yeah, we did that for in the real data part. Like we find some co-founders and then we kind of correlate with something known. Like say for example, LDR T2D example, we were testing whether other co-founder represent other lipid level, HDL or triglyceride total, or glucose level, or so we did check them. So in general, it's hard because those we can check whether. Essentially, we can check these co-founders because you don't observe co-founder. Only thing you know is the G2U effects. You can infer G2U effect and say whether kind of match any known factors, any known kind of risk factors. Yes, most often it's hard to match, kind of have a good match. This factor generally kind of
makes sense.
More subtle than what you can kind of actually measure.
Makes sense. Sorry, Daisuke.
So okay. 37. Okay.
31. Okay. So okay. So this is a bit biological question I had. So if you find the factors which are not known, then what are you going to do next? Maybe a biologist or a medical scientist needs to go to do some more experiment, or can you go do something more to do some statistical test?
That's a kind of general question about MR. So the initial, the motivation of MR exactly do that, finding some risk factor of cancer or T2D, and then and then prove it using, say, randomized clinical trial or biological studies, molecular biology, to understand whether it's a true effect, and then maybe that become drug target. That's how LDR become. So LDR is essentially a lot of focus on LDR on heart disease come from earlier MR studies. So it's very influential. So, and we have people, including ourselves, trying to find more cases like this, because that's really the major impact of treating a lot of common disorders. But in practice, it's using MR alone is not enough, as I kind of said. I think cause cannot completely avoid false positives. So in practice, not usually enough. So I think the hope is to give some hints, and then someone will follow up.
Yeah.
Can I ask, very relevant right here? I think that Daisuke and I kind of getting similar questions. But Shin, if we look at like traditional GWAS studies, right? So you would have defined or identify 100 risk loci, and then according to these type model, what percentage of them do you think come up as false positive?
GWAS, in GWAS, most of them are probably rare if you use strong threshold, like 10 to the minus 8 p-value. So most of them can be replicated. So you use an independent study recruiting totally different patients, usually are associated replicate. So the false positive rate actually very low there.
So there you think that the false positive rates are, okay, I see.
So that's for GWAS, but not for Mendelian randomization, because Mendelian randomization makes pretty strong assumption, as I explained. But for GWAS, most loci, if you find it, they're probably rare.
Okay.
Yeah.
But they could be like, well, those are just correlation, right? So.
Oh, if that's what you mean, no, they are not. They are real in the statistical sense, but not in the biological sense. They may not be causal variant. I guess that's what you mean.
Right. That's what I meant. That can the model now like separate out the causal variants from the GWAS and the non-causal variants?
Yeah, that's something we have been working on. We use technical phi mapping. So it's like a sparse regression, like for people who have worked on lasso. Like if you look at the region of a human genome, you already have many variants that are all associated with Y for your phenotype. So you essentially got Y equal to X beta, where X is many correlated variables. So you can imagine you can use something like a lasso where choose a small number of variables to explain all the data, right? So those things are more likely to be causal. So those similar ideas are being used because of phi mapping. Even though kind of lasso is a good analogy, we don't use lasso because lasso kind of doesn't guarantee to give a causal variant. We use some kind of Bayesian approach to this selection, variable selection. Yeah, that's a really another interesting question. Yeah.
Okay. So any other question?
We're running out of time. If you are interested in some of those things, and you feel free to email me after the seminar, I guess.
Thank you, Shane. Let's thank our speaker one more time. It was a great, clean talk. Thanks so much.
Thank you very much.
Very nice.
For staying with me. Thank you.
So Shane, I'll stay behind just to chat a few more minutes, if you don't mind, just to catch up.
Yeah, absolutely. I'd like to talk to you more.
Sure. That's okay. So this was, by the way, our last seminar of this semester, right?
Yeah.
So we'll pick it up hopefully next semester from the beginning of fall semester. And that's okay. We'll chat probably to set up like a
schedule, right, like a
so
time and so on. Yes.
Right. All right, sounds good. Let's see.
Do you have a different Zoom budget or?
No, this is fine. So I think that we'll stand a few minutes. Or Daisuke, would it be fine if we use the same Zoom?
Yeah. So this is fine. This is fine. So you two are the host. So you can keep this.
Thank you so much. I'll see you soon, Daisuke. Thank you.
Okay. Thank you.
Bye. Thanks.
Bye.
Bye.